The final step, oxidation, which is the breakdown of these fatty acids. Oxidation, as I have told you, that it is of three types: beta, alpha and omega. First we will discuss about the beta oxidation. Beta oxidation, there are 4 steps of this beta oxidation. For example, here we have a molecule of fatty acyl CoA, active fatty acyl CoA in the mitochondrial matrix, which is like this. CoA portion. Now, first of all, why it is called as beta oxidation? This is carbon number 1, 2, 3, 4, 5, 6, 7, 8 and so on. This one is, for example, I am going to explain the beta oxidation for a 16 carbon compound, because carnitine shuttle is involved for the very long chain fatty acid. So that's why I am going to explain with the reference of 16 carbon compound. So this is the carbon number 16. This process will take place, or we can say that the splitting of this fatty acyl CoA molecule will take place at the beta carbon. With each process, with each beta oxidative pathway, there will be removal of 2 carbons, or in other words, you can say that our fatty acyl molecule will become shorter, or shorter than before, 2 carbon shorter than before. Like, for example, if for the reference I write it with another color like this. This is the carbonyl carbon, and next to the carbonyl carbon is the alpha carbon and beta carbon. Now, the splitting will be at this point. So, if the splitting is taking place to the beta carbon, so that's why, because beta carbon will be the last carbon. So, that's why this process is named as beta oxidation. And in the beta oxidation, the first step is dehydrogenation reaction, or the enzyme involved in the first step is the dehydrogenase enzyme. Like this. Like this, you can see. So, there will be the formation of double bond. There will be the formation of double bond. This is fatty acyl CoA, fatty acyl CoA, and the enzyme involved is fatty acyl CoA dehydrogenase. Enzyme involved is fatty acyl CoA dehydrogenase, and there will be the formation of FADH2. There will be the formation of FADH2. Once again, listen to me that in the first step, fatty acyl CoA through a dehydrogenase, with the help of dehydrogenase enzyme, is converted into another compound, which is named as 3-enoyl CoA. There will be the formation of 3-enoyl CoA, and also there will also be a formation of FADH2 molecule. So, after this, what about this dehydrogenase? The dehydrogenase, it has 3 isoforms. It has 3 isoforms. If we talk about the dehydrogenase, it has 3 isoforms. Number 1 is G phase. G is a green colored, we can say the isoform, and this is specific for the fatty acids having a chain length of 4 carbon to 8 carbon. The second one is Y1, yellow colored flavoprotein dehydrogenase enzyme, and this one is specific for the fatty acid having a chain length of carbon number 4 to carbon number 18, and it is more specific for the fatty acids having a chain length of 6 carbons. And the third one is Y2. This one is specific for the fatty acids having a chain length of 6 carbon to 16 carbons. 6 carbons to 16 carbon, and it is more specific for 16 carbon fatty acids, more specific for 16 carbon fatty acids. And here, as we are discussing about the oxidation with reference to 16 carbon fatty acid, then the isoform of the dehydrogenase involved is Y2. That clear? And its full name is fatty acyl CoA dehydrogenase. Is that clear? And it will be responsible for the formation of 3-enoyl CoA. Now here will be the addition of 1 molecule of water, and as a result this double bond will break, and here will be a hydroxyl group, and here is CH2 by the addition of 1 molecule of water. What is the mechanism involved here? Here if you see, so try to understand and listen to me very carefully, that I am just going to explain, draw only this part of this fatty acyl CoA. Like for example, we have this structure, and also we have water. We know very well that this water will be converted into H positive and OH negative. So what will happen, that with the help of that enzyme, what changes will occur? Here the electrons will be transferred to one side of carbon, and as a result we will have like this. The electrons are transferred to this side, so here will be a partial negative charge, here will be a negative charge, and here will be the positive charge. Now according to the chemistry, the OH in the water, which is negatively charged, it will attack on this positively charged carbon, and the hydrogen in the water, which is positively charged, it will attack on the carbon with negative charge. So you can see in the diagram, CH, one hydrogen will attack on the CH and it will be converted into CH2, as you can see, and OH, when it will attack on this positive CH, it will be converted into CHOH. Now this compound is named as 3-hydroxy-acyl CoA, because all the process is taking place through the third carbon, beta carbon. 1, 2, 3, 1, 2, 3. So 3-hydroxy-acyl CoA. Is that clear? And the enzyme involved here is hydratase. Full name is 3-enoyl CoA hydratase, and its older name is crotonase. crotonase. And in the next step, again, the dehydrogenase enzyme is involved, and this portion will be converted into another carbonyl carbon. It will be converted into another carbonyl carbon like this. As you can see. So, two hydrogens, one from here and one from here, these will be removed. If we talk about the chemistry of this reaction, let me show it to you. For example, this is the portion that I have shown it over here. From here, this hydrogen will leave, and as a result, carbon will have a partial positive charge, and this hydrogen will also leave, and this oxygen will have a negative charge like this. Now, this extra electron which is showing the negative charge, it will be shared by the oxygen to neutralize or to finish this positive partial positivity of this carbon. Once again, listen to me, that when this hydrogen will leave, it will have a partial positive charge. When this oxygen will share its electron, which is we can say that which is in the negative charge form, which is in the form of negative charge, it will be shared by the oxygen toward the carbon, and there will be formation of a double bond, as you can see over here. As you can see over here. Is that clear? And as a result, this whole molecule will be named as 3-ketoacyl CoA. It will be named as 3-ketoacyl CoA, and the enzyme involved is dehydrogenase. And again, dehydrogenase is involved, so there will be the formation of one molecule of NADH2. After that, the last step of the beta oxidation, that is the cleavage with the help of enzyme thiolase. With the help of enzyme thiolase. With the help of thiolase enzyme, this fatty acyl CoA will split itself at this point. So there will be the formation of CH3 C double bond O and the second molecule, and thiolase enzyme will add one portion of CoA, as you can see in the diagram. Now, this is again a fatty acyl CoA, this is again fatty acyl CoA, and this molecule is named as acetyl CoA. So this is the complete beta oxidation, and now this fatty acyl CoA is shorter, 2 carbon shorter as compared to the parent molecule, as compared to the original molecule. This fatty acid was having 16 carbon, but after the beta oxidation 2 carbons are removed, and the remaining fatty acyl CoA is of 14 carbons. Is of 14 carbons. Is that clear? So in this complete beta oxidation, the enzymes which are involved, that is dehydrogenase, that is hydratase, again dehydrogenase, and again the thiolase. These 4 enzymes, actually these are involved in the beta oxidation. So this is a 4-step reaction. This is a 4-step pathway. Is that clear? And once again I am going to repeat that in the first step, with the help of dehydrogenase, this fatty acyl CoA is converted into 3-enoyl CoA, and with the help of hydratase, enoyl CoA is converted into hydroxyacyl CoA, and this hydroxyacyl CoA with the help of dehydrogenase is converted into the ketoacyl CoA, and ketoacyl CoA with the help of thiolase is converted into acetyl CoA and fatty acyl CoA, and its mnemonic can be FAHC. Its mnemonic can be as you can see FAHC. Its mnemonic can be FAHC. F for fatty acyl CoA, E for enoyl CoA, H for hydroxyacyl CoA, and K for ketoacyl CoA. And after FAHC, we will have 1 molecule of acetyl CoA and again fatty acyl CoA. Is that clear? This was the beta oxidation. And in the beta oxidation, we have 3 types of the dehydrogenases: G, which is green colored and specific for fatty acid having 4 carbon to 8 carbon; Y1, which is specific for fatty acid having 4 carbon to 18 carbon and more specific for 6 carbon fatty acids; and Y2, which is specific for 6 carbon fatty acid to 16 carbon fatty acid and more specific to the 16 carbon fatty acid. And this beta oxidation that I have explained to you, it is with reference of palmitic acid, which is a 16 carbon fatty acid, and I have told you that in each beta oxidation there will be the removal of 2 carbons from the fatty acid, and as a result the new fatty acid will be 2 carbons shorter as compared to the previous one. Like here 16 carbon fatty acid, but here at the end of beta oxidation this fatty acyl CoA will be of 14 carbon and 2 carbons are removed. This process will go on until unless we have only at the end we have only the 2 carbon compounds. Once again listen to me, that this beta oxidation process will go on until unless we have left only 2 carbon compounds at the end. Then the beta oxidation will stop, because there will be no more fatty acid, or there will be no more long chain fatty acid more than 2 carbons or more than 2 carbons. Is that clear? So this was the beta oxidation.